Okay, so welcome back to the workshop. We're on day 7 now. So just before we begin the session, I just want to just make a quick announcement that once again we don't have any formal group meetings today. But we all encourage you to please come and meet with the mentors, myself, Chitabra, Mithun, Somideep, Soling, and Harish. During Yeah, sorry? Mithun will be coming a bit later today, but the rest of us are all here. So please do meet with us during the tea breaks and the lunch session if you have any questions or if you would like any feedback regarding your projects. Yeah, and you can yes, please meet with the speakers as well. Okay, so with that, let's begin today's sessions, and we'll begin with the second talk from Professor Ajishman Raju on a Waddingtonian view of development. So please go ahead.
Okay, thanks, Shakti. So this is not the original title of the talk, so I changed the title, and I decided I'm going to talk about, also because of 1.5 hours, so there's some time. So I'm going to talk about two different two different projects, the first one is going to be what I was earlier going to talk about, where I'll build on some of the things we talked about yesterday, and tell you about the how we think about, in a particular example, in early mouse development. I'll show you how we use some of the concepts that I built up yesterday as a tool to quantitatively understand what's going on in a particular cell fate specification process that's happening in early mammalian development. The second part of the talk is going to be on something a little different, and it's going to essentially bring in some ideas from evolution. So I'm going to talk about, really again, look at some early ideas of how development was seen as important for evolution, which was subsequently revived, and again tell you how we thought about them, tell you about some of the relevant experiments, and again how we are thinking about it in the kind of framework that I'll tell you about. Okay, so let me begin. So the first part is going to be on modeling cell fate specification in the mouse blastocysts. So that's early mouse embryos, and this is work in collaboration with Eric Sigia at Rockefeller. So just to remind you, some of the things that we talked about yesterday is that there is a parallel between ideas from development and ideas from dynamical systems theory, right? So in general, you would write down differential equations for change of gene expression with time. On the right hand side, you would have some functions which will tell you how production and decay terms. And as we talked about yesterday, the way to visualize this is through these kinds of diagrams, where you have these stable fixed points which correspond to differentiated cell types at the relevant time scales. You have these kinds of saddle points. So we didn't get to discuss saddle points yesterday, but we did discuss unstable points. And saddle points are essentially points where some directions are stable, other directions are unstable. So you can see this, this cross here, it has a stable direction and an unstable direction. These saddle points are points essentially where the cell has to decide whether it's going to become one type of cell or the other type of cell. And then you'll see these domains of stability, and these correspond to what would be called specification. So again, these ideas of decisions, differentiation, and specification come from developmental biology, and these ideas of fixed points and domains of stability come from dynamical systems, and there's a nice overlap between the two. Okay, so of course, one of the issues that we discussed yesterday is that typically when you're thinking about these decisions in development, you're dealing with very, very large dimensional systems, right? You have signaling pathways which have many components. Even at the transcript level, you have many genes involved, and it seems as if the more we study these systems, the more complex they seem to be. But ultimately, I think there's a belief, at least, that dimensional reduction is possible, right? And that's given by these kinds of diagrams that you will see in many places. So this is for zebrafish, where they've done single cell RNA sequencing during development at different stages of development. And right, this is quite common. You'll see these kinds of two-dimensional diagrams which try and cluster different types of cells. And so the very fact that you can take this very high dimensional data, which is the gene, at the RNA level, the gene expression in all of the cells, and you can visualize it in a two-dimensional plot, is evidence of the fact that you think that there's some sort of dimensional reduction is possible. That really the underlying dynamics is taking place in a smaller dimensional space, and you don't have to pay attention to the large dimensional space in which the actual dynamics takes place. And so, this intuition, this idea that dimensional reduction is possible, actually goes back to early experimental embryology. And in particular, Waddington, who was in the talk title, had this idea that you can imagine developmental dynamics as a landscape, right? So here I'm showing you a figure from his book, The Strategy of Genes, and so this is all early days, this is 1957, but he's imagining genes to be pegs like these, and there's a network of interactions here, and this network is essentially controlling some sort of landscape. And the landscape you should think of as being on top of the landscape means you're not specified, and as you roll down in the landscape and go into one of these several minima, these minima correspond to differentiated cell types. So this is also a picture that's become very common now with the advent of stem cells, and to quote from Waddington himself, he said that it is important to realize that the comparatively simple orderliness of the epigenetic landscape, he called this epigenetic, meaning above the genes, is a property of a higher order dependent on an underlying network of interactions which is vastly more complicated. So again, this is in 1957, so he already had an idea that you can have a vastly complicated network of interactions, but you can get emergent simplicity. And the way he thought of the emergent simplicity was in terms of this kind of a landscape. And in drawing such a landscape, he was trying to build in certain biological insights, in particular the idea of canalization. And canalization essentially meant that this landscape had this very particular structure where you had these kinds of valleys and if you like, canals, which were really forcing your cell to go on a very specific path. And I'll discuss this a little bit also later, that it's not when you perturb the dynamics on the landscape, you don't just end up at an arbitrary place. You really can only go along one of these pre-specified paths and end up at one of these minima, all of which correspond to differentiated cell types. So he was bringing in the intuition that we discussed yesterday, that there are a few discrete types of cells, even though the state space may actually have more possibilities. In the end, differentiated cell types have a few discrete types, and these minima are, for him, these minima corresponded to those kinds of cell types. Okay, so essentially what we're going to do is build on this early intuition, and what we want to now formalize it and use it as a mathematical tool. So we don't just want to use the landscape as a picture, which is just giving you sort of, it's not just a metaphor, but we really want to take the landscape seriously as telling us quantitatively what's happening to the cell when it's taking a decision. So again, I'll set up some mathematical preliminaries, but very soon I'll go to a specific example where hopefully all of these concepts will be illuminated. Yeah. Yes. The dynamical is the higher dimensional description. So this is the totally general description. Yeah. And then, yeah. Yes, exactly. Yeah, that's the idea. Okay. There are a couple of concepts that you need to go from the idea of the general class of dynamical systems, just a bunch of differential equations, to an idea of a landscape. And the first one is this idea of what's called structural stability. And what structural stability is that if I take a certain set of differential equations and I perturb them in some arbitrary way, I just add whatever term you want to the differential equations, does it change the qualitative behavior of the differential equations, right? And remember, yesterday we discussed the idea of qualitative behavior through this idea of topological equivalence, right? And so there is the idea of a qualitative behavior of a differential equation. The question is, if I take, you give me your favorite set of differential equations which you think tell you about the gene regulation, you perturb it with some arbitrary functions, does it change the qualitative behavior of the differential equation? Does it change the number of fixed points? Does it change whether a fixed point goes from stable to unstable and so on, right? And if it doesn't, then you say that that is this concept of structural stability, right? And so René Thom was somebody who initially linked the idea of structural stability, again a mathematical idea, with the idea of robustness and canalization. That you can make perturbations, but in the end actually you end up, the general qualitative nature of the equations doesn't change so much. It turns out that there are a class of mathematical systems whose mathematical properties I'm not going to go into, they're not so important, called Morse-Smale systems that have this property. The important things that you assume in this is one, the most important is structural stability. You assume that whatever differential equations you write down have this property that small perturbations don't change the qualitative behavior. And the second is actually that the flow is bounded. There's another assumption you have to make, which just means you don't escape off to infinity, which is a biologically reasonable assumptions, right? You're talking about chemical concentrations, you don't just escape. They don't become infinity. In the end, something saturates it, and so eventually the flow is bounded. So these are the two assumptions, and that, essential assumptions. And you can show that Morse-Smale systems are systems which behave those assumptions. They're structurally stable. And further, it turns out there's a theorem that these kinds of systems can always be written as a potential plus a metric. For this whole talk, I'm actually going to discard oscillations. Oscillations make things more complicated, so you again, you mathematically prohibit oscillations. You also, structural stability means you can't have things like chaotic behavior. And again, the idea is in biology, you don't want your system to become chaotic. You don't want it to be on the edge of chaos where a small perturbation is totally, not in all of biology, but in development in particular, you want something that's robust, that's canalized. So mathematically, you get rid of, oscillations of course are important in many contexts, but just for this talk, I'm not going to be talking about oscillations. So no oscillations and no chaos. So essentially, you are just left with these ideas of stable and unstable fixed points in your dynamical systems and bounded flows, the kinds of things that we were discussing yesterday. And essentially, what Smale showed was that these kinds of systems can always be written as a potential, which means as a landscape, plus a Riemannian metric. For this talk, again, I'm going to forget the Riemannian metric. If people are interested in discussing this with me later, I'm happy to. But this slide is just to show that, in fact, mathematically there is a way to way to make Waddington's idea of a landscape precise. That really you can show that a certain class of differential equations can be written as a gradient flow, can be written as a landscape. So that's, as far as we knew, this wasn't, this theorem hadn't been noticed, but in any case, it's very powerful, for it tells you that, essentially you can use a landscape, literally a landscape, mathematically speaking a potential, to model, the biological system that you're interested in, where the cell is differentiating, no matter what the underlying equations are, the gene regulatory equations are. So that's something that we are going to be using as I go forward and tell you about the example. Yeah. So in general, so again, I could write it, for example. If I have some equation like this, this is my equation. So in general, a small perturbation means any function on this side with some small parameter. And if, yeah.
Overall, it's not changing the sign, or we can say.
So the question is, does it change the qualitative behavior of the equation itself?
Yes.
And those are the kinds of questions you're interested in.
So, like I can say, if adding this in the
Yes.
equation
Yes.
will not change the sign, but a little change in magnitude will happen.
So what do you
Can
mean
we
by sign?
Positive or negative, like the direction will not be changed, but magnitude will change.
So again, remember we are talking about differential equations in large dimensions. So in principle, you could also have other variables. And if you want to visualize them in terms of flows, then the direction is really in some high dimensional space. So you can change the direction also, you can change the magnitude also. What you can't change is the fixed points, you can't change the number of fixed points, you
Steady
can't change the
states.
signs of the eigenvalues near the fixed point. Their stability you can't change. So that was the idea of qualitative behavior. But as such, you can change things about the actual flow. Yes.
Okay.
Yes. Yeah. Finite. Yeah. It's not for infinite dimensional systems. Yes. Yes. It's the same, though this is a distinct theorem. Yeah. The Smale theorem is a distinct theorem. Smale did a bunch of things. Yeah. Yeah.
Can you elaborate in the biological context? What exactly is the flow?
Yeah, so I'll get to that in example, but you remember yesterday the flow was, right, if I have some gene regulatory network, right, I had some protein concentrations as a function of time. The flow was telling me how those protein concentrations change as a function of time. The figure I showed you here was RNA, but essentially gene expression as a function of time flows, that just means that it changes as a function of time, and that change has a direction. Yeah. Huh?
Any intuitive idea how the metric is coming?
Sorry, I couldn't hear you.
Any intuitive idea how the metric?
Oh, how the metric? So yeah, the metric is essentially saying that the flows. So there are two ways to this. So it's essentially saying that the flows are not downhill in the potential. Really, the flows can be more complicated. Another way to think about the metric is that if I have a gradient flow, then near any fixed point, nearby, if I linearize near the fixed point, I'll only get a symmetric matrix because my flow is derived from a potential. So my linearization near the fixed point will be two derivatives of the potential, which will be a symmetric matrix. In general, of course, near a fixed point, I can get any, I don't need to get a symmetric matrix. So the metric is needed in a sense. The metric is another symmetric matrix, but the product of two symmetric matrices can give you a general matrix. So that's why the metric is needed. It's really needed to, the potential keeps the fixed points. The potential is the minima. It's really, if they have three cell types, then the potential better have three minima. The metric can change the flow going from one cell type to another cell type. So it really can change the dynamics. But again, I'm not actually going to use the metric for this talk. Yeah, so I'm just going to think about potential flows where you really have a landscape and the landscape determines for you your equations of motion. Flowing down the landscape corresponds to what's happening to the protein concentrations. Okay, so one more bit and then I'll go to the example. And that is, of course, that you're interested, as we talked yesterday, in these kinds of bifurcations. You're interested in changing an external parameter in a way that can change things qualitatively. Like, so yesterday we saw that an external signal can actually, destroy a stable fixed point, and we had two stable fixed point and unstable fixed point, and as we change an external parameter, one of them was removed. And so you would jump from low protein concentration to high protein concentration. So those kinds of bifurcations you want to allow. And you want to allow them because in the biological context, you want to allow perturbations of signals that are determining the decision. So experimentally, the fact that I can perturb signals means that bifurcations are allowed. And so I have to also build in the possibility of bifurcation. So first, what I've shown you is that you can model the dynamics using a potential, but in that potential, you need to allow bifurcations, which means some of the fixed points go away, some of the minima disappear, other fixed points might come. And then it turns out you can ask what kinds of bifurcations are allowed. I won't go into this, but basically there are only two kinds of generic bifurcations. Generally speaking, unless you do something special in your equations, there are two kinds of bifurcations which are important. One is what's called a saddle node. That's exactly what you saw yesterday, where a point like this and a stable fixed point, they annihilate each other. And so I've changed a parameter from here to here. These are the flows in some two-dimensional system. And as I change a parameter, suddenly this stable fixed point disappears. Initially the flows were attracting here, now the flows will take me here and then bring me either here or here. The second kind of bifurcation, which we didn't talk about, but which turns out to be important, is called the heteroclinic flip. And what the flip does is, the reason it's important is, again, I want to think about all this in terms of cells differentiating. So I want to say, if suppose I'm in this state, and then I'll go into this or this state, which will correspond to different cell types. When I exit this state, maybe with a bifurcation, I want to know whether I'll reach the left or whether I'll reach the right, right? And the heteroclinic flip is a bifurcation which precisely changes that. So if I change things a little bit again, then here I go directly to this point, and I'll go left or right only using noise. Whereas in this thing, again, I've changed one parameter slightly, I'll go to the right directly. If I have a bifurcation of this fixed point, my flows will take me to the right. So that's another bifurcation that we need to consider. Yeah.
Fixed points.
Yes.
So these are signaling. So how important could be fluctuations? Because fluctuations can really take you to different fixed points. And given that these are signaling, number of molecules involved could be less.
Yes, yes.
So should
the
one
fluctuations are important. So in what I show you, there will be noise always.
So these are all deterministic dynamical systems. So
yeah, deterministic dynamical systems, and we do nothing very special about the noise. We'll just add like Langevin noise or something like that.
Okay, so you do look at the effect of noise.
Yeah, but you'll add these. So let's just say we don't think very deeply about the noise. The deterministic dynamical system sort of sets up the framework, and then we add noise in it because, as you're saying, of course, there are fluctuations. Yeah. Sorry, I couldn't hear you.
Can you again explain what a heteroclinic flip is?
Yeah. So so a heteroclinic orbit is an orbit which connects two points like these. These are called saddle points, again, because they're attracting in one direction and they're unstable in the other direction. And you see there's an orbit, there's a flow. If I start from this saddle point, I go directly to this saddle point. This orbit is called a heteroclinic orbit, a heteroclinic connection. If I perturb, so there's a very special kind of orbit, right? I'm going from this saddle point exactly to this saddle point. If I perturb things slightly, I destroy that orbit. Now if I leave from this saddle point, I'm going to go here. I'm not going to go to this saddle point. I could have perturbed it the other way, so that again I destroy this connection and I go from here to the left hand side. So that's important from the biological point of view, because I want to know if I escape this cell state, where will I go? Will I go to the left or will I go to the right? So that's why the flip becomes important. Yeah.
By signals you mean perturbations, right?
So I mean signaling proteins. In the mathematical context, there will be parameters, which will be which will perturb these equations. Yes. I don't know if what you meant.
The question here, same as fluctuations, right? But I thought like signals are like these change in parameter, some fluctuations and all, so that they are changing this landscape.
Yes.
So, but what I asked, I got to know like what he asked. Like
Yes.
I understood like signals and these fluctuations are different.
That signals are?
Signals and these signals and those fluctuations are different.
Yes, yes. So they're slightly different because noise is right. Noise is sort of so signals are things that can change the landscape, right? You should really think of it as things that can signals control the landscape and can change it. Fluctuations are the fact that cells can be in different parts of the landscape but can move around noisily. We won't think of fluctuations as changing the landscape. We'll think of fluctuations as the fact that cells move around noisily in the landscape, whereas signals we think of as actually changing the structure of the landscape. So a fluctuation would never get rid of a fixed point in this framework. Yeah.
Thanks.
Yeah.
So this heteroclinic flip, can it be can it be can it be constructed by like changing the network as if like of the system that we are studying? I mean,
It can be constructed by changing parameters in the network. Yes. If that's
yeah, I mean like so this is due to the signal, like some change in the signaling that is causing this flip, right? But I'm saying like suppose you have a network,
Yes.
and it has a, let's say you're modeling it as a through a dynamical system,
yes.
and it has this, let's say this right now it's like a heteroclinic kind of an orbit is there.
Yes, yes.
Yeah, but let's say I'm changing like the the network as if let's say, so can it lead to this flip that you're talking about?
So yes, so the mathematics is actually agnostic to what you choose to change, right? Biologically, the signals are what you can often perturb. Of course, biologically you can also make, right? You can knockouts and so on, so you can also do mutations which are not just signal perturbations to the signal. So the mathematics doesn't care what you choose to change in your differential equations. So these kinds of bifurcations can occur whether you choose to change the decay rates, whether you choose to change signals, external signals, right? So again, you can get these based on changing the network, unless you meant by changing the network somehow changing the state space itself. For example, removing a node. This can be a little bit more tricky because then you're changing the state space itself. But if by changing a network, right, for example, you mean removing a link, that's effectively like setting a parameter to 0. This doesn't care. So you can get bifurcations that way. You can get it in any other by changing any parameter in your equations, in your differential equations.
So is it like something? So the like you're changing the separatrix over here when you're doing the flip over. So the separatrix is changing by like in the case of the left-hand one, there is a like a distinct separatrix is there which is separating the two flows, right?
Yeah.
And in the third
Yeah,
case,
I mean, so there will be an orbit that also separates the flows here, right? This orbit, for example. So everything to the left of this will flow here, and I mean, so there'll be some orbit that will also separate flows here. But the point is that the connection between the two is lost. If I escape this fixed point, I am now in the region of stability of this fixed point. I am not in the region of stability of this fixed point, which is a separate orbit here. Right, it's only this region. So that's what the bifurcation is doing. And again, biologically, it's important for the reason that if I change, if I'm a certain cell state, if that changes, I want to know where will I go. Will I go to cell type A? Will I go to cell type B?
so given the map that you have, can you just re-engineer the let's say the network so that it can give rise to this kind of map? Like what I mean, let's say you have a dynamical system, then you can get this map, okay. But let's say you have a map, can you like re-engineer the system?
So that's what I want to avoid. I want to say that actually there will be many networks which correspond to this map, and I want to get away from thinking about the networks and instead think about the potentials. The potential is an effective emergent description. The landscape is an emergent description. I want to get away from thinking about the networks. There are actually many networks which will do this, many ways to do this, but since those are quite complex, instead we look at a simpler effective description which just looks at the potential that arises because of the network. So I would say that there's not a one-to-one map from the networks to the potential. In general, there are many networks which will give the potential. Yeah.
Okay, thank you.
So yesterday you showed like for positive autoregulation we see bistability which
Yeah.
can give memory.
Yeah.
So I mean, you said already like many networks can give this feature, but what could be a simplest two signal mechanism that gives this feature?
I mean, so again, I want to get, so the reason I want to avoid this is because I want to appreciate, I want to bring about appreciation for a new philosophy, which is that we should get away from the networks because the networks are very complex. In any case, the equations for the networks you don't know. They are, any case, phenomenological equations, as I was saying yesterday. So it's true, you could try and think about a few components, and I'll show you in the example also, which can give you this kind of dynamics. But I want to sort of try and bring appreciation for a new philosophy that if you don't know about the network in particular, then you can still find out a lot of things about biological interest which don't have to do with the specific network. And this is what Waddington's insight essentially was, that actually the fact that you get discrete cell types is telling you something important about the dynamical system. And so there's a shift in emphasis from the network to the potential. So which is why I want to. So let me maybe as I go through the example, I'll show you how this is a useful way to describe things. So I've already told you yesterday that the task of dynamical systems theory, right, is to break up your you have certain parameters in your system, right? As you change parameters in the system, again, the mathematics doesn't care what you change. Experimentally, there may be certain parameters you can access easier than others. But as you change parameters in your system, the task of dynamical systems theory is to tell you, draw a bifurcation diagram, which will tell you what are the different kinds of regions, qualitatively distinct regions possible in the dynamical system. And it turns out that, right, so for example, if you're changing two parameters, you could get regions like this. So regions where you have these three stable fixed points, there's a saddle node bifurcation here, so you get rid of this fixed point, you only have two stable fixed points. You have a heteroclinic flip here, so again, the flows go to the left, here the flows go to the right. So these kinds of bifurcation diagrams tell you essentially all you need to know about the dynamical system. And the important result that sets the basis for the work that I'll tell you is that actually there are only a few of these types of bifurcation diagrams that are possible. In particular, if you take systems with three stable fixed points, then again under very, very general assumptions, right, assumptions I've told you about, structural stability, and the idea that suppose there are two things you can experimentally perturb, two parameters, whatever you have access to, there are only a few classes of bifurcation diagrams that are possible. So it's a very, very powerful theorem. It says that you take any dynamical system you want, any network you want, okay, and you change any two parameters of that dynamical system. If that dynamical system has three stable fixed points, then whatever the dynamical system is, it will the bifurcation diagram will fall into one of these few classes that we enumerate. Again, of course, the dynamical system satisfies certain of these properties. It has to be structurally stable and so on. If it doesn't, then you can get more complex things. But this is really in the spirit of universality, in the sense that you don't care about the molecular components that are shaping your dynamical system. Effectively, what you know is that you're going to get a few discrete number of cell types, and you can enumerate the kinds of bifurcation diagrams that are possible as you vary any two parameters of your system. So we're going to use this sort of powerful idea and see how it works in early mouse development. So I'll go through, I'll go through this example in a little bit of detail so that some of these ideas become clear. Okay, so we are talking, so in early mouse development, you have this, you have a zygote, initially you have this cleavage, you get a few number of cells, and then the decision making happens through two binary decisions. The first decision is between what's called the trophectoderm and the inner cell mass. The second decision is what's called the primitive endoderm and the epiblast. We're going to be interested in the second of these decisions, the inner cell mass going to the primitive endoderm and epiblast. The epiblast ends up becoming the actual embryo proper. The primitive endoderm and the trophectoderm actually make supporting tissue. Okay, so we are going to be examining this decision. Again, you can see how it fits into the framework that we have. You have three types of cells possible: the inner cell mass, the primitive endoderm, and the epiblast. We are going to be examining this time scale where this decision takes place. Eventually, of course, these things differentiate into, there's further differentiation, but we are only going to be looking at the time scale where this decision takes place. And now we are interested in using this sort of general mathematical framework to see what we can say quantitatively about this decision. Okay, so the first thing that it so happens in this way, in this case we know the signaling protein that can change the number of primitive endoderm or epiblast cells that you get. So it turns out to be the FGF protein. So the adding overexpressing FGF leads to all primitive endoderm. So if I treat this thing with a high dose of FGF, then I get all primitive endoderm. If I treat it with an inhibitor of FGF, then I get all epiblast. So again, right, as I'm changing a biological signal, I'm not getting new types of cells. All I'm doing is I'm changing the proportion of cells that are primitive endoderm and the proportion of cells that are epiblast. So again, this tells you something interesting about the underlying dynamical system. As I change a signal which enters in some complicated way, it's transduced and then further enters the gene regulatory network. I don't still get new types of cells. I only permute between known cell types. So it falls into the kind of framework that we've been discussing. Okay, so here's what a typical model would look like, where you would try and say that there's an underlying network. There are two genes involved, GATA6 and NANOG, which repress each other. There's an ERK signaling pathway. There's some signal transduction. The FGF is received, goes through some signaling pathway, and then it it maybe activates the GATA6 or inhibits the NANOG, right? And so the kinds of differential equations that I have not been writing are differential equations like these, where you would have variables for all of these components, and then you would have some guess for what the right hand side is, which typically is Hill's functions and so on. Again, to mention, this is a simplified gene network model. Actually, these signaling pathways are far more complicated, but you think that this sort of captures the essence of what's happening in this system. So this is from a sort of existing model, and I'm going to be talking about changing the way we think about this decision in two ways. The first one is, instead of now this network description, we are going to have a potential description. We are going to have a landscape description of how this decision is taking place. And the second is something that I actually avoided talking about so far, but which now I have to talk about, which is that so far we've been talking about decisions that take place in a cell, right? But actually, of course, in this there are many cells, and these signals, in this case the FGF signal diffuses, and so that it diffuses, and you can see all of these cells can take these kinds of, right, they can all become either primitive endoderm or epiblast. And so existing models, what they would assume is that these signals are local, and so if I have I have one cell which ends up becoming epiblast, then my neighboring cells, the signals will only reach the neighboring cells, and the neighboring cells will tend to adopt the other fate, right? So I'll show you this diagram again. So here initially you have all inner cell mass, you start getting blue and red. The idea is that if I am blue, then my neighbors will sort of be red, right? And that's because there's some signaling going on between cells, and if I am blue, I sort of make my neighbors become red. And what that assumes is that the signal is spatially localized, that the signal doesn't travel very far, and my blue cell signals and makes my neighboring cells red. And this is, so these are the two assumptions in the sort of existing model. One, you have some gene regulatory network, and second, you have spatially localized signals. So the first part is that I'm going to replace the gene regulatory network with a potential, and the second part actually relied on data, analysis of some recent data that became available. So recently there was some live imaging data of the ERK activity. So remember, ERK is a proxy for the FGF, because the FGF signal goes inside and goes through the ERK pathway eventually to take the decision. These GATA6 and NANOG are markers for the fate, epiblast and primitive endoderm. And so there was live measurements of the ERK. So the ERK was measured as a function of time, which allowed us really to now check at single cell resolution. So it allowed us to check whether this idea of spatially localized signaling is correct. Essentially what we found was that there's not, if you had spatially localized signaling, then in the kind of model I described to you, if I am a certain type of cell, my neighbors will be different. That would mean that there would be an anti-correlation between the ERK activity of one cell and the ERK activity of its neighbors. And we did find a small anti-correlation, but essentially as you can see by the spread here, this anti-correlation is very small. And what this pointed to was that actually the signal was not spatially localized, and it was not that your neighbors were very strongly anti-correlated with the cells, that the cell's neighbors were very strongly anti-correlated with it. And the reason this was happening is basically that there are two reasons. One is that these cells are actually moving around. So in this early blastocyst, these cells actually move around, and here this is a 2-hour period. So this decision takes place over about a day. This is data taken from a 2-hour period. The center of these spheres are where the cell starts. This whole thing is the blastocyst. This is a cavity that forms. As you can see, these cells actually move around quite a lot, right? That means that if I had a neighbor at a particular point of time, I may not have the same neighbor after 2 hours. And in fact, we could sort of check this, and we found that every half an hour the cells actually switch a neighbor on average. So that means there's no meaning really of spatially localized signaling, because in any case you're switching around neighbors all the time. These cells are moving about. Of course, the signal is also diffusing. That's the other thing. The signal is diffusing. So the signal is diffusing, the cells are moving around, which means that we can really think of this as some sort of well-mixed system, where the signal essentially becomes some sort of average homogeneous signal. Yeah, so this I already talked about. The cell changes neighbor every 30 minutes, which we quantified. So now I'm going to put together these two ideas. One, there's the signal is like this some sort of well-mixed system. Second, instead of the gene regulatory network, we should think of the potential. And so this is the cartoon of how we want to think about this, right? So we assume the FGF to be homogeneous and uniform. So it's not actually homogeneous and uniform, but it moves around enough, the cells move around enough, and this FGF diffuses enough that effectively we can think of a homogeneous FGF, homogeneous signal. And second, we don't model the genes, we only model the cell fates. So only the cell fates are modeled. There are 3 kinds of cell fates. The cartoon version of it is really that you make a potential. This can have these 3 kinds of fate: inner cell mass, epiblast, and primitive endoderm at some reference signal level. As there's high signal level, you end up, the potential changes, the signal changes the form of the potential, and you can become the primitive endoderm fate here. So this is the cartoon, but is essentially how we're thinking about modeling this kind of decision. And as you can see, it's actually a far more simplified description of what's going on. Okay, so really how do we think about this decision? And it turns out there are 2 kinds of ways that this decision can happen. There are 2 kinds of landscape you could draw, right? One of them is a landscape on the left, where initially your inner cell mass here in yellow, and then you take a decision either to become primitive endoderm or to become epiblast. You decide to either flow to the left or you decide to flow to the right. So those are the 2 possible decisions, right? Inner cell mass to primitive endoderm or inner cell mass to epiblast. A distinct way to take this decision is the one on the right, where initially your inner cell mass, first you decide to be something that's not inner cell mass. So you flow down, and then you take a second decision whether you want to be epiblast or primitive endoderm. So you can see the flow diagrams here are distinct. In one case, there are 2 saddle points here, so you either go from here to here or here to here. In the other case, you first escape this fixed point, and then there's a second saddle, and then you decide whether to go left or the right. These are topologically distinct. So this for example, all the fixed points are connected in a 1-dimensional curve. Here you have really, it's a distinctly 2-dimensional topology. And so these are sort of the 2 ways that you could imagine taking taking this decision. One is you go from ICM to primitive endoderm, ICM to epiblast. The other is you go from ICM to not ICM, and then you take a second decision whether you're going to go to primitive endoderm or epiblast. Okay. So I'll pause a second if this is clear. Yeah.
Why do you say this is topologically different? Because I can see there are 3 stable fixed points and 2 saddle points. So I
Yeah.
can move around the saddle points, right?
Yeah, so this now is really a little bit something we haven't talked about, but it's also about how they are connected to each other. Not just the fact that they are there, but also how they are connected to each other. So in this case, right, they're really all in a straight line. They're, you go from here to here to here to here to here. There's a sort of straight, not straight line, I mean, a single one-dimensional curve which connects all of them. In this case, they're not, in this case, you cannot do that. This topology is essentially two-dimensional. And as I'll show you, it also ends up leading to different kinds of bifurcation diagrams. So it's true what you're saying, something that we haven't discussed, but these are in fact distinct. I mean, the other thing you can notice here is that there's a heteroclinic connection here, right? In this kind of a topology, there cannot be a right? There's no heteroclinic connection here. There's just there's a stable fixed point in between. So those are reasons why actually these are distinct ways too. But intuitively also, you know, the way to think about it is that it's these are distinct ways in which you can take the decision. Yeah.
So see, in the first case, for the cells to take a decision to differentiate into
Yes.
either of the types
Yes.
from the initial, the ICM basically.
Yes.
So you said that they have to take a decision to either flow into the first, one of the fixed points, right?
Yes,
So
yes.
is that decision making entirely noise dependent? Because say
Right.
I start from a particular the beginning fixed point and I
Right.
have to flow into either of them.
Right.
So I can only imagine it to be some sort of a
Yes.
noise which would push them into either of the
Right,
fixed points.
right.
Is it only because of that when you talk about it in a biological
Yeah,
context?
yeah. No, that's a great question, and it's something that one has to decide whether you flow from here to here purely because of noise, or is there some other signal which leads to a bifurcation here. And it's a little bit related to the timing of these decisions. So it does turn out that actually these decisions take place asynchronously. Not all the cells decide when they want to become one or the other. In fact, in time, this is sort of asynchronous. It happens, you know, the cells decide at different times. So that would seem certainly that it's noisy, right? It's not deterministic. There's certainly noise has a role to play. On the other hand, the initiation of the decision also seems to be quite specific, and it really seems to have to do with cell number. And so then that would seem to suggest that there is also something that's controlling the timing, something that's actually so far not biologically understood. And there are different ideas for what could be controlling the timing, which would then suggest that actually it's something that's leading to a bifurcation. So what we decided to do was so first of all, we decided to use this topology rather than this, for reasons I'll tell you about. And second, we decided to create a bifurcation but also have noise there. So both of them are happening, but you don't purely escape by noise. In the end, you escape because this fixed point just disappears. You just bifurcate away this fixed point, which is a timing mechanism in this decision. So there's a timing mechanism, but there's also noise. So both of those things are happening.
So is it right for me to say that to make this more deterministic, the landscape has to go through some sort of a heteroclinic flip, so that you can direct more cells to become to reach one particular cell fate more than the other?
Right. So that's so precisely what we will do. But that's to control something which I haven't mentioned to you, but which I will. I guess I can do it right now. I mean, so the other phenomena here is that you end up getting this ratio of cells. So you have about order of 50 cells. 60% end up being primitive endoderm, 40% end up being epiblast. And this ratio turns out to be very, very stable. You can do all sorts of things with the embryo. I'll even show you some experiments. And you still seem to get this 60-40 ratio. There's this extreme robustness at the level of how many cells give you primitive, how many cells are primitive endoderm, how many are epiblast. And so to model that, what we have to do is to say that the the signal itself, now remember the signal is homogeneous, it's acting on all the cells, but the value of the signal depends on the state of all the cells, some average state of all the cells. So if for example here half the cells have somehow managed to reach this part, then my signal, my internal, so my signal is also produced by the cells, right? Then my signal compensates to now tilt the landscape. If you're deep inside the well, it doesn't tilt it enough that you'll escape the well, but all of the rests, you know, the rest of the half which are still here waiting to be decided, they'll all flow to the left, right? So, so maybe in the flow diagram it's clearer. If I start with say 50 cells here, 25 of them flow to the blue state, then my signal, the way the model is set up is if 25 of the cells are in blue state, then my signal compensates so that the landscape shifts in this way so that everything else will go to the green state. So that allows you to maintain this kind of robustness where the signal is dependent on the state of the cells. So here's where the heteroclinic flip becomes important in deciding how many cells will take one fate versus the other kind of fate.
So is this sort of a setup experimentally observed where?
Yeah, so I'll show you the experiments.
Yeah.
I mean, so I don't know which part you're saying is experimental.
Where the landscape is tilted based on the cell states which you're talking.
So the landscape is not a direct experimental observable.
Yeah.
Right. The fates are observable. The what fates the cells take, this is an experimental observable, and the fates you know, the landscape makes a prediction for the fates, and you can fit the fates using the landscape. So that's the
by experimentally observe, what I meant was say you produce that when these you said that when the cells are in a particular state, they produce some sort of a signaling factor which could. So is that production of the signaling factor observed?
Yes. So it's it's true that cells only produce FGF if they are in the epi state.
Okay.
So this certainly the epi fate is correlated with the production of FGF. Yeah. So that is definitely. Yeah.
So when you talked about tilting the landscape, for example, when 25 of them would reach there, it will then the signal will say change to tilt the landscape on that side so that it becomes skewed for other cells to go that side. Can we connect it to spatial localization of signals that neighbors are perhaps telling the other cells to have reduced gene expression?
So that was the old model, but here the signal is homogeneous. Every cell sees exactly the same signal. So there's no spatial information in the signal at all. And that comes from our analysis of the spatial correlations of the ERK signaling in the cells, where there don't seem to be any strong correlations. And so the signal is assumed to be homogeneous. So all the cells see the same signal, but it's sort of, it's sort of a self-adjusting potential, if you like, where if things become too heavy on one side, then the potential adjusts to make things biased towards the other side.
So in this case, what is telling the system that it is becoming heavy on one side?
So the fact that some cells have changed the state, which has led to greater production of the signal, that signal has diffused and homogenized. I mean, also the cells are moving around, and so now you have more of the signal, and that has told you that actually the rest of the cells now see more of the signal. That pushes you to the other side.
Sure.
Landscape is this form of the potential, is it?
Huh?
The landscape is the form of the potential, is it?
Yeah, exactly. Yeah.
Details of this potential are derived, or you are taking some simple form consistent with what you see.
Right. So you take a simple form which is consistent with this this kind of where the fixed points are, and then polynomial potentials are enough. So you just simple polynomial potentials suffice.
So then the mechanism, the potential changing depending on the population at this, those things cannot be derived.
So the mechanism is parameterized as coefficients of the polynomials. So the signals have to.
But time evolution I'm talking about.
So the time evolution is just the gradient of the potential. So
when the potential itself changes, there should be a time dependent mechanism which changes it.
So
that has to be derived from more microscopic description, I guess.
I mean, so there's a potential. The gradient of the potential is the flow, and then you're right. So the potential has parameters, right? these parameters are functions of time.
Yeah, exactly.
So these parameters are functions of time in a phenomenological way, yeah. I mean, so you have to remember, I mean, in this case, the microscopic description to begin with is phenomenological. There is no underlying deep truth here. Microscopic equations are in any case phenomenological. So this is doing phenomenology in the sort of simplest way you could, in the really keeping the essential characters that are involved in this, yeah.
And what makes the percentage 60-40 you mentioned that very robust? What makes it robust?
Well, what makes it I mean, so in our description, what makes it robust is this kind of construction. What makes it biologically robust, is that what you're asking?
Yeah.
I mean, this is a harder question. I mean, this is presumably the product of evolution, that you have this kind of setup where, that has created this landscape in the first place. These landscapes have presumably been sculpted through long years of evolution. But how you get that, this is a harder question, yeah.
So I'm just wondering, what is the use of the landscape? How are you going to use it?
So I'll show you. Yeah. So I'll show you how we use it. Okay. So as I said, so now these bifurcation diagrams in this case are different. So again, remember the bifurcation diagrams are regions which tell you what kinds of fixed points exist. So for example, here there's an ICM epi, there's a tristable region where all 3 fixed points are there, and there's some monostable regions and so on. There are some interesting features of this which I won't go into. All I want to say is that these 2 kinds of potentials have different kinds of bifurcation diagrams associated with them. Okay, so first I'll show you now first how you can use these potentials to fit existing experimental data. Let me also tell you how you could experimentally distinguish whether it's the first kind of landscape or the second kind of landscape. Right, and so again I'll show you a thought experiment, but again I can discuss later. These experiments are not so hard to do. Imagine that you have certain cells that start at the ICM, which is near a bifurcation, and you bias your system in some way so that it starts flowing towards the epi state. In this case, it's the blue state. And then as these cells all of the cells start going to the epi state, at a certain point of time, you give a dose of FGF signal, which will take things to the primitive endoderm state. Remember, high dosage of FGF takes you to the primitive endoderm state. In the first kind of landscape, here there's just a flow diagram associated with the potential. You have to go back to the ICM state to be able to go to the primitive endoderm state. There is simply no trajectory which goes directly from here to here, which you can see also in the potential. There's no trajectory which goes directly from here to here. You have to go back to come here. Whereas in this kind of a potential, you have a direct route to go from primitive endoderm to ICM, and so that's clear here. You may be biased towards the epi state, but you can be easily pushed to the primitive endoderm state. And so this is why we prefer this kind of a potential, because it gives you more time to decide once you've exited the ICM state. It's easier to push. It gives you more time to whether you want to go from one or the other primitive endoderm or epi state. So that's the sense in which this is now used very, it's in quotes. I don't mean robust in any quantitative way, but this is why we prefer this kind of a landscape to try and model this phenomenon. Another experiment which actually again you can do is to realize that different parts of the trajectory have different sensitivities to the FGF. So again, if I have a cell here which was started at the ICM, then it moves about noisily and reaches the epi state. If I perturb it early, if I give an early FGF signal, the perturbation does nothing. So this is my perturbed state in red. I perturb it, then again sort of comes down and reaches the epi state. If I perturb it a little bit later, the late FGF pulse actually changes the fate. And this is a small pulse of FGF that can change the fate if I perturb it at the right time. That means that when I give a pulse of perturbation of my signal, will have different effects in both of the landscapes. And this I'm showing you here. In the first kind of landscape, the sensitivity to the signal just declines with time, and if you've studied the developmental biology, this is the idea that potency declines with time, that initially you're very potent and then as time goes on you lose your potency. This is a distinct idea. This says that actually you're most sensitive at intermediate points of time. So initially when you're at the ICM state, you're actually less sensitive to the signal, and it's only later on that you become more sensitive to it at some intermediate time. So again, this is a, you can do these kinds of perturbations giving pulses of the signal, and then this would give you different results depending on what is the underlying landscape. So there are ways in which you can experimentally distinguish using these kinds of perturbations of the signal which kind of landscape is controlling the decision. And then the next thing we do is, of course, fit these landscape to existing experimental data. So I'll describe to you the kinds of data that exist. The first one is what I've already told you, this idea of robustness. So when wild type cells are mixed with GATA6 mutants, which end up making only the epifate, they adjust and give rise to primitive endoderm. Right, so here, so these are like these, you were seeing these chimera experiments yesterday. Basically you label the cells. So these are, by the way, these things are very, the reason people study the mouse blastocysts is because you can manipulate this outside of the mother, right? So these are, and in fact it seems like you can do whatever you want with them, and they're still, till the blastocyst stage, the decision making happens very robustly. So again, if you take wild type cells, you combine them with these GATA6 mutants in different ratios. The wild type cells, which if you just had all wild type, you would get 60 PRE, 40 epiblast. If you combine them with other cells that are primed to be only epiblast, then the wild type cells self-adjust. They start giving only primitive endoderm, so that the ratio remains 60-40. So that's, an experimental, these are experiments that have been done, and that of course the model has to predict, as in model has to fit. And then there are other kinds of experiments. So there are experiments where you can put in the FGF for the whole time. So this happens between 2.5 days to 4.5 days of embryonic development. You can put in FGF, you can put in the inhibitor of FGF, you can put in FGF for some time, then the inhibitor. You can grow them in FGF and then in control media. So you have all these kinds of perturbations which give you in the end fates of cells, whether you get primitive endoderm or epiblast, and so that all of this data we fit to. This here is the experiment that I just showed you. Depending on the percentage of wild type cells, you can plot the fraction of PRE cells. If there was no regulation, you would expect a straight line here, but there's clearly a lot of regulation, which is this robustness that I already talked about. And again, this you can compare the experiment and the model. And finally, there's other data just showing you how the decision takes place. What fraction of cells are ICM, what are PRE, what are epiblast, and this, as I told you, happens asynchronously over this whole period. So all of these are, experimental data that's available in the literature. The fits here are actually almost trivial. You just have two parameters in your potential. There are a few other parameters you need to bring in to have the signaling and so on, but it's sort of the minimally parameterized model you could make. If you imagine taking the kind of model I showed you before and fitting the same kind of data, which has not been done, you would have 74 parameters in that model. So this is really decreasing the complexity of the model as far as possible, and still giving you results that fit to a good extent existing experimental data, and also make predictions about what will happen when, for example, you do kinds of experiments that haven't been done, like giving a pulse of FGF. And they show you something interesting about the way the decision takes place, that instead of losing potency in time as you would have thought, you actually have this intermediate regime where you're most sensitive to the signal. Okay, so I'll stop here about this, and then I'll move on to the second part in the last 20 or so minutes I have. But if there are any questions about this, then I can take them.
In the heteroclinic flip, you told that the ICM first becomes something that's not ICM, and then differentiates into pre. What does it become? Something not.
I don't know. The state doesn't have a name, but it's. I don't know of a better name for it. But what I mean is that just in the gene expression space, I guess, you. Yeah, I don't have a good name for it. So I don't know what would be an answer to what does it become. Do you think, are you asking does it become a different type of cell?
Yeah, of course it doesn't become a different
Yeah.
type of cell,
Yeah,
right?
yeah.
So is it being modified in some other way? Because
Yeah.
I just didn't get the concept of the heteroclinic flip, because ultimately you have a cell and
Right.
it ultimately differentiates into the pre-endoderm or the epiblast.
Yes, yes.
You don't have a mid stage in the middle, and that's what we are assuming in the heteroclinic flip.
Well, so in a certain sense, you always have, again, remember, if you're just looking at the level of protein concentration, so I agree, so this is the way the diagrams are drawn, that you have inner cell mass and primitive epiblast. But if you think in terms of the protein concentrations, right, the markers here would be nanog and GATA6. So if I tracked, if I imaged nanog and GATA6 expression, then I would get some continuous trajectory going from double positive nanog GATA6 states to either single nanog high or GATA6 high, which would be markers for these states. So you do have a continuous expression, a trajectory that goes from something that you would call ICM to something that you would call epiblast or primitive endoderm in the level of protein concentrations. Now the question is, what kind of dynamics does that trajectory, what kind of dynamics does that trajectory take? And I was telling you that there are two possibilities for it. Now you could ask, I mean, what are the, what should I think of this as nanog and GATA6? I mean, that you can't do because again, we are not modeling the genes. We don't have a network, we are not modeling. So we are just modeling the cell fates. So in principle, this space is some reduced dimensional description of the full gene expression space. The kinds of descriptions that you would want to do in any case that often are done more for aesthetic purposes. So there is some reduced dimension in which the dynamics is taking place, where not everything matters. And there you can ask, well, how could it, what could be the trajectories? And so these are distinct ways for the trajectories to take place. And now what these axes are, for us the axes are abstract. In principle, the axes are some combination of gene expressions, some nonlinear combination of gene expression that we are not modeling here. We are only modeling the fates, whether you get one or the other kind of fate. Yeah, but there are midway states in the sense that there are protein expressions that are midway between a cluster of cells, cluster that you would call ICM and a cluster that you would call primitive endoderm and epiblast.
So in the other model, the dual, the old model, in that we don't assume that the protein concentrations are varying. We just assume that one cell directly becomes the other cell.
No, they vary here too. They vary here, they just vary differently in the sense that you only take a decision, right? So they still vary. So there's still a continuous trajectory that goes from here to here. It's just the trajectories are different, they're distinct. And the way they're distinct, I was trying to show you with the signaling perturbations, can show you how they're distinct, right? So here, in one case they stay on this line, in the other case they actually move about in this two-dimensional thing. And so that has some experimental consequences. Yes, but in both cases they have.
And you also said that in the heteroclinic flip, the pre-endoderm and the epiblast, they're connected. So you mean like 60-40, the ratio is balanced. That's what you mean by?
No, by connected I meant that there is a way, so I meant that this thing in the potential. There's a way to go directly from here to here. There's a trajectory which goes from here to here, whereas here there's no way to go directly from here to here. You have to go up and then come back down. Yeah. Of this, so this, right, so precisely this, that if I bias things to go in one direction, and in between, so first I bias things to go in one direction, and then I hit it with a signal that actually biases it in the opposite direction, my cells will have to come back. So like these kinds of plots that I had, right, where ICM is here, so there's again existing experimental data where you take gene expression, you classify cells as ICM, primitive endoderm or epiblast. Now I would predict that if you do the experiment where first you bias things to go towards the epiblast and then you bias them to go towards the primitive endoderm state, which are not experiments that are different from this, but at the same time as doing this experiment, you also dynamically keep track of how many cells you classify as ICM, how many you classify as primitive endoderm, how many you classify as epiblast. Then I would see, sort of ICM going down, epi coming up, and then ICM would go up again a little bit, because again you have to go back up, you have to go back towards ICM, right? Whereas in the other case, once you've lost ICM, you've lost ICM. You're no longer going to get ICM expression. So if there was in these kinds of experiments, only the end fates were measured. the gene expression was not dynamically, the markers were not dynamically measured. In these kinds of experiments, there were no perturbations. If these two were combined, the model would make a prediction for what, how these curves would look. And in one case, things are going towards epi, it seems like, and then you push it with a signal back to PRE, but to do that, you have to come through ICM. In the other case, you can just directly go. So ICM would always monotonous, it would just keep declining, it would never come back up. Yeah.
Since I don't have a very strong mathematical background, I'm just trying to build an intuition.
Yeah,
so
yeah.
Just let me know if I got it right. So cells have a particular gene network which would help you determine this particular landscape. And yesterday we, for example, saw that when you change the external signal, it changes the topology of the particular state, right?
Yes.
So again, so the quantities of the components of the genetic network determine where you are in the flow diagram, and the landscape is changed by factors like the external signal here, for the example. So would it be, so if I extend this to a later developmental context, say there are regions where this external signaling component is different in amount, so each region would have a different landscape, which would in turn say can give rise to different topologies in different regions, which in turn would give rise to different stable points, meaning different cell types. So would that be a right way to think about this?
No, so that's a good question. And again, it goes to the question of, right, so far we're talking about the landscape. In fact, I'm going to just come to that in my next part. So far we're talking about landscape at the level of single cell. And again, if you see all of the stem cell literature, right, it's all single cell, how it's differentiating. But now the question is, if I have many cells, is what does the landscape mean? Does every cell have its own landscape and then things are going on? Or is, is there some, in this case, I got away with it by assuming that essentially the cells are independent and there's a homogeneous signal.
Right.
If the signal has some complex spatial character, then you have to think about it more, you have to then, problem becomes a little bit more complicated. So that's something maybe we can talk after this about how you handle those situations. Yeah.
Let's assume there are 3 cells.
Yes.
The A cell, A, B, C. So
3 cell types,
yeah, 3 cell types. Not exactly 3 cell types. It's the first stage of being cell type. For being
Sure. Yeah. 3 kinds of cell states that are you're interested in.
So if A is releasing something, like you already mentioned, that neighbors affect the cell type of the other cells, right? So A is releasing compound A and C is releasing compound C. Now what about the B cell, which are intermediate between A and C cells?
So in this case, so first of all, the neighbor thing is, what we were finding was that model doesn't quite hold. That the neighbor idea is not correct because neighbors are switching around and signal is diffusing. But second, in this case, the ICM is not a signaling state. In this case, only the epi state is signaling. So only when you're near the epi state, you signal and push things toward the primitive endoderm state. There could be other more complex cases where you can have signaling that depends on all 3 states. And so there's been some work, for example, in C. elegans, where the situation is made simple by the fact that there are few number of cells, but you have 2 types of signal there. It turns out you have notch signaling and you have EGF. And again, these signals now, you again think of it in terms of a landscape. You have 3 fates. And then depending on where you are, you have to decide how the signaling takes place. So that's something that you have to model, how being in the cell state affects the signal. And then the signal affects the cell state in turn.
Like if you are in the intermediate position of 2 different cell states, then what could be the final cell stage of that intermediate cells?
What could be? I didn't understand the question.
Final fate of that intermediate cells.
So the final fates will always be one of these few fixed points. So in the end, we actually, to assess fates, in the end you basically, right, so cells lose potency. So eventually you in any case stop responding to the signal. So in the end, the diagram is just, you just have certain regions, and depending on which region you are in, you adopt that fate. So if you're in some intermediate fate, you will eventually just flow to whatever fixed point you're near, which will correspond to being in that cell state. So in the end, there are only 3 possibilities. Was there a question? Okay, fine. Okay, so then let me just quickly, I have not that much time, but I'll maybe quickly just give you a flavor of the second part of what I want to talk to you about. Maybe I'll skip these conclusions. Okay, so so the first, as introduction, so now this is actually now going a little bit into thinking about these landscapes from a different perspective. And so far, as I said, we've been thinking at the level of cells, but really actually if you go back to Waddington's original work, he was not drawing these landscapes for cells. In fact, he was thinking of these landscapes as really corresponding to robustness of whole phenotypes. And that's very interesting. So this is now work in collaboration with Bing-Kan Shu at Florida and Stan Leibler at Institute of Advanced Study. Now we're going to start thinking about, we're going to take the analogy in the direction that Waddington took it in, which was to think about the landscape for whole phenotypes, for not just for not just at the level of single cells, but really as there being robustness and canalization at the level of phenotypes. So to give you an example how that might work is what you've heard about a lot here is Turing patterns. So it turns out you can think about Turing patterns also in this kind of a landscape way, where essentially initially you have an unstable fixed point, which is your homogeneous state, spatially homogeneous state, and then you flow towards a stable fixed point, which is your patterned state, and the space in which you think you have a landscape description is now now the Fourier space, the space of modes that you heard about yesterday from Philip Maney. But in that space, it turns out you can also write down a landscape description for Turing. Yeah.
In the equation for SOX 9,
yeah,
where is the cubic term coming from?
Yeah, so these equations are not mine. So these equations are taken from this paper, and the way they model the equation. So the point of this paper was to show that you don't just, and the way they decided to model the equations was, the way Gierer Meinhardt did it was really to say activator inhibitor and try and make some equations for activator inhibitor. They decided that they'll only make the equations near the homogeneous fixed point. So they assumed you're near a homogeneous gene expression state. Near that gene expression state, you have a linear part, which are these linear equations, and then you saturate away using these kinds of cubic terms. So these are all phenomenological. There's no deep.
Any higher order odd term would
saturate it. Yeah, exactly. So that's why they decided if you don't saturate it, you escape off too.
I see.
So you need to saturate it. That's why. Oh, sorry. They put this cubic term. Yeah. Right. Anyway, so these. So this work I'm not going to talk about, but work that we are doing to show that these three-component Turing systems also you can think in terms of a landscape, which is work that my student Shubham is doing. But there's actually a very interesting debate between Crick and Waddington, which was all, so there's Francis Crick and Waddington, they actually wrote these letters to each other, where, Crick was basically annoyed with this idea of a landscape, and he was discussing with Waddington, what is this landscape, what does it represent, and these letters actually get quite, quite angry. They get quite, so what Crick says is, I had expected you to say, was that the path of the ball represents the lineage of a single cell. So he was imagining this path again, right, ball rolling down landscape and taking a final state as something happening at the level of a single cell. And then Waddington says, for heaven's sake, why? Why do you think it's just confined to a single cell? Actually, I can think of this ball as representing larger phenotypes or patterns and so on. And so for Waddington, this was really a description that went beyond just single cells, but really was the fact that development was canalized in the sense that you really got discrete phenotypes, it seems like. And how that happened, to see this, to understand this, in particular, he did a bunch of experiments. And so I'll just quickly tell you about the kinds of experiments he did. I'll skip this slide. The first experiment that he did was to look at the fly wing. And you actually heard a lot about the fly wing and how it patterns from Amitabha. What Waddington found was that if you heat shock flies 20 hours after the pupae forms, so if you remember from yesterday's talk, that is exactly when these wings are forming, it leads to a break in the posterior cross vein of the fly. Right, so you get these breaks in the posterior cross vein, and the interesting thing was that breaks that he was getting from heat shocking the flies as they were developing were also to be also known mutants. So these environmental perturbations were phenocopying known mutants. There's a crossveinless mutant of the fly which also showed a break in the posterior crossvein. And he could obtain exactly the same just by heat shocking the fly. So heat shock is a very arbitrary perturbation, right? So he heat shocked the flies and got the same kind of phenotype. So again, very interesting thing that you take an arbitrary environmental perturbation and you phenocopy a known mutant. For him, it was more evidence for the landscape. The fact that at the level of phenotypes, there are only discrete possibilities. And whether you push it there with the environment or you push it there with the mutation, it doesn't matter. There's an effective landscape that's telling you the possibilities that exist. What he did next was he actually did these artificial selection experiments where he took the so not all the flies got a break in the posterior crossvein. There are a percentage of them that showed the alternative phenotype. He took the percentage that showed the phenotype, then he did artificial selection, right? He selected only those flies and he created a new generation, and then he kept doing this till he got 100% penetrance. So that now he had a bunch of flies that he would heat shock them 20 hours after the pupae form. All of them would show this kind of a phenotype where there's a break in the posterior crossvein. The interesting thing that he observed was that after a certain number of generations, about 13 or so generations, he could get flies that showed a break in the posterior crossvein without a heat shock. This phenomena he termed as genetic assimilation, that a character that was initially environmentally induced as a result of an environmental perturbation could be genetically assimilated in the sense that it could be expressed it could be expressed in the absence of the perturbation after a few generations of artificial selection. He then did this in a more vivid example where he actually exposed early fry embryos to ether, and that turns out to phenocopy the bithorax mutant, where these flies actually get these two pairs of wings. So there's a very it's not a small break, it's this very abrupt phenotype. And again, the point of this was, again, this was a known mutant. So again, the point of this was that something that's ether, you're putting in ether in an early embryo, has the same effect as known mutation. So again, for him, all of these were evidence of the fact that underlying development there is a canalized landscape, and you can push things in the landscape either using environmental perturbations or using mutants. There's been much discussion of what kinds of molecular changes could be involved in these, what kind of molecular mechanisms could be involved in getting this. I mean, there's been a lot of discussion around HSP90, heat shock protein. There's been some suggestions it could be transposon activated mutations. Waddington's own idea was that essentially there were polygenic changes, that again there was some, right, there's this landscape that formed. There are polygenic changes which allowed you to express this new phenotype, but the important thing was that the structure of the developmental landscape had this specific character where environmental perturbations don't just allow you to get crazy mutants, they actually give you things which you already, which are already known mutants and which seem to be very, very specific in nature. What we did was we went through the sort of set of experiments in that have been the set of experiments that have been done over the years. I mean, his initial experiments were done in '53. Since then, there have been several, times his experiments have been repeated. And we essentially classified the results of these experiments and summarized them using these 4 curves. One was this increasing penetrance. The one was this assimilation curve, the fact that around generation 15 or so, you start getting genetic assimilation. Without the environmental perturbation, you see the new phenotype. The other two I'll ignore for now because we're running out of time. The kind of theoretical model that we needed here was again to, so again we want to think about it in terms of a landscape, but really now we can't think of just a few fixed points and a signals, we're not looking at a very specific developmental decision, but we wanted to capture something that more abstractly brought out Waddington's conceptual insights. And there are many of them. So in particular, the important ones are his idea of homeoresis. So again, it's a term he invented. Homeoresis was the dynamical counterpart of homeostasis, which I'm sure you'd all heard of. And it was the idea that you have trajectories which are stable, dynamical trajectories that are stable, not just homeostasis is sort of like fixed points. Homeoresis is the dynamical counterpart of that. This idea of branching, that it seems like developmental trajectories have different branches, and if you perturb them, as I said, either using environmental or genetic perturbations, you choose a different branch. And then finally, in our model, we wanted something that was consistent with the experimental curves. So, we basically, this ended up being an exercise in almost a separate exercise where, there's a known model in physics for how you store fixed points in some general dynamical systems called the Hopfield model, and we wanted to sort of generalize that and think about something where you could store dynamical trajectories and not just fixed points. So that's something we did. I won't go into the details of it. We're almost out of time. Let me just show you sort of how Waddington thought of it and sort of our corresponding pictures. So his idea was initially, you have this landscape. Initially, you go to this fixed point, which corresponds, let's say, to the wild type. The environmental perturbation pushes you over the hill right here and takes you to a different trajectory. This could be the cross veinless or the bithorax, depending on the nature of the perturbation and where during development you are. And then finally, genetic assimilation meant that the landscape itself changed, and the ball naturally rolled down now to this other phenotype, to this alternative phenotype, which he was able to get with his selection experiments. So what he was trying to argue using these experiments was an important idea that in thinking about evolution, you have to take into account development. And the fact that actually evolution is acting on phenotypes means that there is this intermediate between the genotypic space and the phenotypic space, which is this developmental landscape. And the nature of this developmental landscape is very, very interesting, and it will have consequences for how actually evolution takes place. So this is something that, of course, so in our model, we tried to make sort of alternative pictures. I'll sort of skip this and just show you, sort of the corresponding kinds of curves that we got for penetrance and assimilation, which is what we wanted. It turned out to be harder than we thought it would be. And I'll just, I guess, conclude and say that, his genetic assimilation experiments, we think, retain interest after 70 years. What we did here in this part that I talked about was to collect and systematize the experimental results and sort of summarize them in these curves to develop a theoretical model. But essentially, in this work in particular, more experimental work is necessary but we just thought it's a very, very interesting phenomenon and brings out some deep insights about biology, in particular the relationship between evolution and development. And of course, there are lots of questions about the relationship to many, many discussions that have taken place on plasticity and evolution and so on. Okay, so I'll just conclude here. I'll stop by letting you know about this TFR, Physics of Life program that we've recently started. We started this last year at NCBS, where I am. And so any of you who are looking to do a PhD, this is specifically designed for people who have a quantitative background with some foundations in math, physics, or computer science, but who are interested in doing things in biology. And so there are many people. You can check it out on the website in the program. It's a joint program between NCBS and ICTS. So I just thought I'd put that there in case any of you are interested. So I'll conclude. Thank you very much.
Yep, please go ahead.
Hey, Chishman, fascinating. So I'm just wondering, like this last thing you showed, is that the epigenetic analog of Hebb rule, like the Hebbian rule that we use for teaching the Hopfield network to embed certain attractors? So could we think of it as something like Hebbian, like the way it's changing?
Well, no, I mean, that's an interesting idea. For us, of course, the learning was taking place through selection. That you are learning these new things through this mutation selection process, which was sort of affecting parameters and taking you to how the analogs it might have to the way we classically do learning. I think it's very interesting to explore.
Yeah, because there is this theory that the evolutionary analog of learning is adaptation.
Right.
And so, but nobody has been, come up with a concrete model for this. So the
Right.
model that you're showing
Right.
seems like the closest candidate I've seen so far of, what's the Hebbian analog for evolution.
Right, right. Yeah, I mean, it's an interesting idea. I mean, the other thing is one part of the model is, of course, which we wanted to keep was this idea that there's dynamical trajectories you have to learn and not just fixed points. But I think it may be true. You can do this, yeah. So you can. I think you can formulate something that's like learning. It would be interesting to compare that with some effective equations for mutation and selection. I think it could be done. Yeah, that's an interesting idea.
And connected to that is this question. This reminds me of the infamous Lysenko affair.
Oh yes.
Yeah. So it almost seems to suggest that maybe Lysenko was onto something, and he wasn't completely talking bullshit, right?
Yeah. No, I completely agree. And in fact, it's written around that time. This is 1950, and it's very, it's titled "Genetic Assimilation of Acquired Characters" for reasons, because the whole controversy was about the inheritance of acquired characters. Right. And what Waddington was placing himself at the center of this controversy, and essentially what he was arguing was that he was arguing what I guess today you would not say is, essentially what he was arguing was you can have this kind of inheritance, but it's not that there's direct inheritance of the environment, but it's more that the selection is acting on this highly constrained space. Which can allow you easily to inherit characters that may otherwise be acquired. And so he was giving essentially a genetic explanation, but I think a creative explanation brought in development. And I agree. I think totally it was, of course that thing became political, so it was taken in a whole different direction. Right. But I totally agree with that, yeah.
Thanks.
Yeah.
Hey, the landscapes that you design, those generally correspond to function, right? The phenotype would correspond to a particular function that is associated with the particular cell, right? So you could have various gene regulatory networks that would lead to same final gene expression, right? As you were mentioning.
Okay.
So gene regulatory networks generally tend to be more modular, right? Like you could have some sense of independence for certain circuits or certain pathways, irrespective of phenotype. So would that also reflect in this sort of, reduced dimensional space? Like would you see modular dynamical landscapes for different parts of the network?
I mean, I don't know. So the problem from my perspective of mapping gene regulatory networks to landscapes is that you have to make certain assumptions about the equations, right? Now, anytime you want to write down an equation, you have to make certain assumptions about what the equations are. And so you can make certain maps. So you can take, for example, particular motifs and say what are the landscapes for these. But those are contingent on particular equations for those motifs. So at least I'm a little worried about making more general statements, but it may be possible to do a certain kind of network analysis which can make more general statements about the topology of networks and the associated lower dimensional description. I mean, in certain cases I've seen things like that, but so I would be, I personally haven't seen anything that can be very general. You can take a specific example with specific equations and maybe try and make some correspondence,
Okay.
Hey, so I'm interested more in the biology of genetic assimilation, which you were talking about.
Yes.
So you were talking about how after introducing some environmental change to a lineage and how the penetrance increased, and after a point where it became inherited, the particular trait became heritable. So was that was it some sort of an epigenetic heritage, or was that heritability epigenetic or genetic in nature?
Possible, I mean, I think it's an open question. So as I told you, the two explanations I've seen from the biology side, one is that essentially you had polygenic changes. So you had sort of very small changes that led you to allowed you to inherit things. which is people who've done you can now, recently they've been repeated and you can sequence, and so and then essentially you don't see any strong, people haven't been able to identify one particular gene that was very strong. In the bithorax case, there is this ultra bithorax. There it may be that actually is one single thing that's very important. The epigenetic, I think it's an open question. I think probably it's involved in some way. The involvement of the epigenetic is these people, a different group, which suggests that actually it's again this transposon activated mechanism. And there you have to take into account other also epigenetic factors. So it's both molecularly, both explanations have been suggested, sort of straightforward genetics versus something that's epigenetic. Part of the reason why we avoided a molecular description is that ultimately you need more experimental work to figure out. Yeah.
Because a genetic explanation seems really absurd to me, because such an environmental change inducing a phenotypic change, and that particular phenotypic change is directly inducing a genetic change which corresponds to the particular phenotype.
Well, so that's exactly what Waddington was addressing, that you would think it's impossible that in 10 generations you can come up with a mutant that gives you the bithorax. But his point was that you don't have to come up with a mutant that gives you bithorax because you think that one gene will give you a phenotype. Actually, different changes at the genotypic level will push you in different trajectories in the developmental landscape. And that landscape is highly constrained, and it's easy, and there are many ways to push you in an alternative trajectory that corresponds to the bithorax phenotype. So it was a different way of thinking about the so I agree, if one takes this whole gene phenotype picture, then it seems totally absurd, and that's why he was right. But he was saying, he did the experiments, and many people are repeating them. So you see the bithorax after 10 generations. So how does it come about? Another explanation, which I should mention, is that it was pre-existing, right? There was pre-existing variation, and you just selected on pre-existing variation.
Right.
So that's possible. Certainly, you do need some pre-existing variation in the population. But as far as I'm aware, nobody's actually taken a population genetics model and actually tried to show it to be consistent with these curves, because most people ignored this for political reasons, because it was too close, so to say, to the so it was totally sort of ignored. These kinds of experiments were done for a little while, but then they were totally
because I'm really surprised that these experiments were done in the late 1900s or something, right? And
no, 1950.
1950, yeah. So and still now, with the amount of progress we've made when it comes to sequencing and everything, I'm surprised no progress has. Little progress has been made.
This is how science doesn't work. So anyway, I'm being recorded, so we'll talk later.
Yeah, thanks.
Okay, so I think if there are any additional questions, we can have them over tea. So for now, let's thank the speaker once again.